You think food poisoning is just an upset stomach and a bad night? Most people do. They chalk it up to that restaurant or that potato salad, something they ate. But there are organisms living in your food right now that can paralyze you, shut down your kidneys, strip your flesh from your bones, kill you within 48 hours. Not in some distant jungle, in your kitchen, on your cutting board, in that chicken breast you just sliced. Every year in the United States alone, 48 million people get sick from contaminated food. 128,000 end up hospitalized. 3,000 die. Those numbers come straight from the Centers for Disease Control and Prevention, and they have not been updated in over a decade to account for population growth. The real toll is higher. In 2024, Paasais, reported foodborne illness cases jumped to 1,392 people sickened. Hospitalizations more than doubled from 230 to 487. Deaths climbed from 8 to 19. 13 outbreaks. Almost all caused by 3 bacteria you need to know about right now. The bacteria do not care about your cooking show tricks or your 5-star hygiene rating. They multiply when you look away. They produce toxins while your food sits at room temperature. They survive boiling in some cases, freezing in others, and acidic environments that would kill most living things. Some need only 10 cells to infect you. 10. A single drop of juice from raw chicken contains enough to send you to the emergency room. This is not about being careful. This is about understanding what you are actually dealing with when bacteria colonize your food, what they do to your body, how fast they can take you down, and why doctors in coastal hospitals keep emergency surgical kits ready during hurricane season, because they know what is coming through those doors when someone cuts their hand while cleaning fish. These bacteria have names: Salmonella, Listeria, Campylobacter, Escherichia coli, Clostridium, botulinum, Staphylococcus aureus, Shigella, Vibrio vulnificus. Each one operates differently. Each one has its own kill mechanism, and each one is sitting in food supply chains, restaurant kitchens, home refrigerators, and coastal waters right now, waiting for the conditions to multiply. Let's talk about what they actually do. If you value documented microbiology and public health history like this, subscribe. It is the best way to support our work. Also, comment from which city you are hearing this from. This script is researched and factual, using sources from the Food and Drug Administration's Outbreak Investigation Database, published February 17, 2025, in the SIDRAP report on foodborne illness increases. The CDC's FoodNet active surveillance data from the 10-state collaborative tracking system established in 1995 tour, and peer-reviewed studies from the journal BMC Infectious Diseases, published in August 2021, on necrotizing fasciitis, mortality rates in coastal hospitals. These are not generic health department warnings. These are frontline epidemiological findings from outbreak investigations, hospital admission records, and culture-confirmed cases. Now back to the information. Start with Salmonella. Non-typhoidal Salmonella causes an estimated 1.35 million infections in the United States every year. That makes it the most common bacterial cause of foodborne illness after norovirus, which is viral. In 2024, Salmonella caused 41 food recalls, up from 27 in 2023 and 21 in 2019. 2 major outbreaks in 2024 tell you everything. In June, cucumbers contaminated with Salmonella sickened 551 people across 34 states. 155 required hospitalization. In January, charcuterie meats caused 104 confirmed cases and 27 hospitalizations across 33 states. Salmonella lives in the intestines of animals, poultry, cattle, pigs. It transfers to meat during slaughter when intestinal contents contact the carcass. It transfers to produce through contaminated irrigation water or unwashed hands. It does not change the smell, color, or texture of food. The bacteria need to reach the small intestine. Stomach acid destroys most of them, but survivors attach to intestinal epithelial cells. They trigger an immune response. White blood cells flood the area. Inflammation. The intestinal lining starts shedding. Diarrhea begins, usually 12 to 72 hours after ingestion. Fever spikes. Abdominal cramps. The illness lasts 5 to 7 days in healthy adults. In young children, elderly patients, and immunocompromised individuals, the bacteria can breach the intestinal wall and enter the bloodstream. Bacteremia. Once in the blood, Salmonella can seed anywhere. Bones, heart valves, meninges. The case fatality rate for Salmonella sits at 0.5%, low compared to others on this list, but with 1.35 million cases annually, that translates to hundreds of deaths. Raw or undercooked eggs are classic vectors. So is raw poultry, unpasteurized milk, prepackaged salads where lettuce was washed in contaminated water. Now, Listeria monocyte genies, this one is different. This one hunts pregnant women, newborns, elderly patients, and anyone with a weakened immune system. Listeria causes listeriosis. The CDC estimates 1,600 people get listeriosis every year in the United States. 260 die. That is a case fatality rate above 16%. In 2024, Listeria led to 65 food recalls, up from 47 in 2022. The boar's head outbreak shows you what happens when Listeria gets into a processing plant. First case identified in late May 2024. Recall issued July 26th. Expanded July 30th. 61 confirmed patients across 19 states. 10 people died. More than 7 million pounds of deli meat recalled. USDA inspectors found numerous food safety violations at the Jarrett, Virginia production plant going back at least 2 years. The plant closed. Boar's Head permanently ceased production of liverwurst nationwide. Listeria does not need much to thrive. It grows at refrigerator temperatures, between 32 and 40 Fahrenheit. Most bacteria slow down or stop growing in the cold. Listeria keeps multiplying. It forms biofilms on stainless steel surfaces in processing plants. Those biofilms protect the bacteria from sanitizers. It hides in floor drains, conveyor belts, slicing equipment. Listeria contamination in ready-to-eat foods like deli meats and soft cheeses is especially dangerous because people eat those foods without further cooking. The bacteria goes straight in. Once ingested, Listeria crosses the intestinal barrier and enters the bloodstream. It has a particular affinity for the placenta in pregnant women. It can cause miscarriage, stillbirth, or severe infection in newborns. In non-pregnant adults, it causes fever, muscle aches, and gastrointestinal symptoms. If it reaches the central nervous system, it causes meningitis, stiff neck, confusion, loss of balance, convulsions. In 2024, the FDA and CDC investigated an outbreak in queso fresco and cotija cheese, 26 identified illnesses, 23 hospitalizations, 2 deaths across 11 states. Clinical illnesses range from 2014 to 2023. The cheese was contaminated during production, probably from environmental sources in the facility. Soft cheeses have high moisture and low acidity, perfect conditions for Listeria. Tomato products, once considered low risk, are no longer dismissed. Listeria adapts. Another ongoing outbreak as of January 2025 involved Yusheng food ready-to-eat meat and poultry products. 19 patients identified. 3 babies died, 2 in California, 1 in Tennessee. 1 baby did not test positive for the outbreak strain and is not included in the patient count. That baby still died. Investigators traced these outbreaks through PulseNet, a national database that tracks bacterial DNA fingerprints from patient samples and food samples. When the DNA matches, they have their source. Move to Campylobacter. Campylobacter caused the most bacterial diarrheal illnesses in the United States. The CDC estimates 1.5 million infections every year, about 20 cases diagnosed per 100,000 people annually, but many more go undiagnosed or unreported. Active surveillance through FoodNet indicates the real number is far higher. Campylobacter jejuni is the main species. It lives in poultry. The European Food Safety Authority reported on 246,571 cases in 2018, an estimated approximately 9 million cases per year in the European Union. In the United States, it sometimes causes more cases than Salmonella and Shigella combined. The infectious dose is low, a single drop of juice from raw chicken, cross-contamination from cutting boards, knives, hands. Campylobacter does not grow in food. It just sits there, waiting. Once ingested, the bacteria head for the jejunum and ileum in the small intestine. They can extend into the colon and rectum. The flagella are essential. They provide motility and help the bacteria adhere to mucus and epithelial cells. Some strains produce a heat, label, enterotoxin, cholera-like, that causes watery diarrhea. Other strains invade epithelial cells directly and cause bloody diarrhea. Symptoms begin after an incubation period of approximately 1 week. Diarrhea, fever, abdominal pain. The illness lasts 5 to 7 days. The case fatality rate is 0.05 per 1,000 infections. 76 people with Campylobacter infections die each year in the United States. But the complications are what make Campylobacter dangerous. Guillain-Barré syndrome, a polio-like paralysis. Individuals with recent Campylobacter infections develop Guillain-Barré syndrome at a rate of 0.3 per 1,000 infections, about 100 times more often than the general population. That is 1 in every 1,000 Campylobacter cases, turning into neuromuscular paralysis. In March 2025, India reported 300 Guillain-Barré syndrome cases. Only about 1 in 1,000 individuals infected with Campylobacter develops it, but India had more than 1,000 Campylobacter outbreaks during 2024. Investigators suspect a specific strain with neurotoxic potential has been circulating. About 20% of Guillain-Barré patients are left disabled. 5% die. Reactive arthritis is another complication. About 3% of Campylobacter patients develop it. Joint inflammation triggered by the immune system attacking the body while trying to combat the bacteria. Symptoms appear around 18 days after infection, can last for months. Campylobacter is mostly transmitted person to person, 60% of cases. Foodborne outbreaks account for a smaller proportion, but are responsible for a substantial number. Raw or undercooked poultry, unpasteurized milk, contaminated water. The bacteria are prevalent in food animals, cattle, pigs, sheep. They are in pets, cats, dogs, birds, farm animals. Any feces from an infected source can contaminate food or water. Young children are most commonly affected, especially in daycare settings where hand hygiene is difficult to enforce. Children shedding Campylobacter in stool may exhibit no symptoms, but remain contagious for days to several weeks. Escherichia coli, E. coli. Most strains are harmless. They live in your intestines, but some strains produce shigatoxin producing E. coli, STEC. E. coli O157H7 is the most notorious. In the McDonald's outbreak in 2024, onions on quarter pounders were the source. 104 patients confirmed across 14 states, 38 required hospitalization. 1 patient died. 4 patients developed hemolytic uremic syndrome, JHUS, a condition that damages blood vessels in the kidneys and can cause kidney failure. Taylor Farms supplied the contaminated onions. Patients became ill from mid-September through October 21st. The company recalled yellow onions on October 22nd. By then, McDonald's had stopped serving slivered onions and temporarily stopped serving quarter pounders at some restaurants. Ground beef tested negative. E. coli O157H7 grows in the intestines of cattle. It does not make the cattle sick. During slaughter, intestinal contents can contaminate the meat. Ground beef is especially risky because bacteria on the surface get mixed throughout during grinding. Leafy greens get contaminated through irrigation water or manure used as fertilizer. The bacteria produce shigatoxin in the human colon. The toxin binds to receptors on the cells lining blood vessels. It enters the cells and shuts down protein synthesis. The cells die. Blood vessel walls break down. In the kidneys, this leads to hemolytic uremic syndrome. Red blood cells get shredded, passing through damaged vessels. Hemolytic anemia, platelets clump, thrombocytopenia, kidney function collapses, acute renal failure. Children under 5 are at highest risk. 1 in 7 children infected with Shiga, toxin-producing E. coli, develops HUS. The condition requires dialysis, often for weeks or months. Some patients never fully recover kidney function. 8 E. coli recalls occurred in 2023. The symptoms start 3 to 4 days after exposure. Severe stomach cramps, diarrhea, often bloody, vomiting. Low-grade fever. Most people recover within a week. But those who develop HUS face a medical crisis. Hospitalization and intensive care, blood transfusions, dialysis. The case fatality rate for E. coli O157H7 infections is low overall, but among those who develop HUS, mortality can reach 5 to 10% even with aggressive treatment. Cooking kills E. coli. Ground beef cooked to 165 Fahrenheit. Leafy greens washed thoroughly, but cross-contamination remains a constant threat. A knife that touched raw meat. A cutting board not properly sanitized. Hands not washed between handling raw and ready-to-eat foods. Staphylococcus aureus does not infect you. It poisons you. The bacteria grow in food and produce enterotoxin. You eat the toxin, not the bacteria. Staphylococcal food poisoning. Symptoms hit fast. 30 minutes to 8 hours after eating, usually 1 to 6 hours. Nausea, explosive vomiting, abdominal cramps, diarrhea. The disease is usually self-limiting. Symptoms resolve within 24 to 48 hours. But it can be severe enough to require hospitalization, especially in young children, elderly patients, or debilitated people. In 2024, a Staphylococcus aureus outbreak at a school in Hebei province, China, affected 61 out of 563 individuals. 10.84% prevalence. Diarrhea, vomiting, abdominal pain, nausea. A breakfast item called rojian mo was implicated. Staphylococcus aureus lives on human skin, noses, throats, hair, fingernails. 20 to 50% of healthy individuals are asymptomatic carriers. Food handlers carrying enterotoxin-producing strains contaminate food through manual contact or respiratory secretions. The bacteria need time and temperature to produce toxin. If contaminated food sits at temperatures between 45 and 118 Fahrenheit, the bacteria multiply. Once populations reach 1 million colony-forming units per gram, sufficient enterotoxin accumulates to cause illness. The toxin itself is heat-stable. Cooking the food afterwards does not destroy it. Foods at highest risk are those that are handled extensively and then left at unsafe temperatures. Cream-filled pastries, potato salad, chicken salad, sliced meats, dairy products, foods that do not get cooked after handling. From 1998 to 2020, the CDC reported 12,139 illnesses caused by Staphylococcus aureus in the United States. In Europe, Staphylococcus was among the top 10 pathogens responsible for the most hospitalizations during foodborne and waterborne outbreaks in 36 countries in 2018. In China, it ranks third highest for food poisoning incidents. 5 typical enterotoxin types exist: C, SEB, SEC, SED, C. Cia is the predominant type, causing global staphylococcal food poisoning, followed by SED and SEB. The genes are located on mobile genetic elements, prophages, plasmids, pathogenicity islands. The bacteria can acquire and lose enterotoxin-producing capability through horizontal gene transfer. An outbreak investigated in France involved chantilly cream dessert. High levels of staphylococci and enterotoxins found in the dessert. Staphylococci also found on a kitchen table surface and in the nasopharynx of 3 food handlers. Prevention requires hand hygiene and temperature control. Food must be kept above 140 Fahrenheit or below 40 degrees. If reheating, temperature should reach 165 Fahrenheit or higher. Refrigerate perishable foods within 2 hours. 1 hour if ambient temperature is 90 Fahrenheit or higher. People with infected wounds or abscesses should not handle food. Gloves help, but do not replace hand washing. The toxin is already formed before you eat. Your immune system cannot stop it. Antibiotics do nothing. The body just has to ride it out, replacing lost fluids and electrolytes until the toxin clears. If you value documented outbreak investigation data and pathogen surveillance like this, subscribe. It is the best way to support our work. Also comment from which city you are hearing this from. This script is researched and factual, using sources from the CDC's National Botulism Surveillance System updated May 21st, 2025. The Medscape Clinical Review on Foodborne Illness increases published August 7th, 2025, documenting the 2024 spike in reported cases, and the MMWR report on the June 2024 botulism outbreak in Fresno County, California, published July 3rd, 2025, involving 8 cases from home canned cactus. These are not summary articles. These are primary epidemiological reports from the surveillance systems that track every confirmed case in the United States. Now back to the information. Clostridium botulinum produces the most potent biological toxin known to science, botulinum toxin. About 1 microgram is lethal to humans when inhaled. Foodborne botulism happens when you eat food containing the preformed toxin. The bacteria are anaerobic. They grow only in low oxygen environments. Canning creates perfect conditions if done improperly. Home canned vegetables are the classic source. The bacteria form spores. Those spores survive boiling. They survive essentially any environmental condition indefinitely. When conditions are right, the spores germinate. The bacteria grow. They produce toxin. You open the jar, you eat it. The toxin goes in. Botulinum. Toxin blocks. Acetylcholine release at neuromuscular junctions. Acetylcholine is the chemical signal that tells your muscles to contract. Without it, the muscles cannot respond. Paralysis. It starts with cranial nerves. Blurred vision, double vision, drooping eyelids, difficulty swallowing, dry mouth, slurred speech. Then it descends. Weakness in the arms, weakness in the trunk, weakness in the legs. Finally, the respiratory muscles, the diaphragm, the intercostals. You cannot breathe. Without mechanical ventilation, you die. Onset of symptoms typically occurs 18 to 36 hours after eating contaminated food, though it can range from 6 hours to 8 days. In June 2024, 8 people in Fresno County, California developed botulism after eating home-canned prickly pear cactus at 2 related family gatherings. All 8 received heptavalent botulinum antitoxin. 5 had botulinum neurotoxin type A detected in serum. Hospital stays ranged from 2 to 42 days. 6 admitted to intensive care. 2 required invasive mechanical ventilation. All survived. The average number of botulism cases reported in the United States is 110 per year. Infant botulism accounts for most of those. Adults get foodborne botulism from home canned foods primarily. Commercial canning uses higher temperatures and pressures that destroy spores, but home canners often do not process long enough or hot enough. Low acid foods are especially risky. Vegetables, meats, some tomato products. The food may look and smell normal. No signs of spoilage. The toxin has no odor, no taste. Boiling for 10 minutes destroys the toxin, but most people do not boil home canned goods before eating. The case fatality rate for botulism dropped from about 50% to 7% over the past 50 years due to improved supportive care, mainly mechanical ventilation and antitoxin. But even with treatment, recovery can take weeks to months. Some patients require respiratory support for months while their neuromuscular signaling mechanisms regenerate. Honey contains Clostridium botulinum spores. That is why honey is not recommended for infants under 1 year of age. Their gastrointestinal systems lack the protective mechanisms that prevent spores from germinating. Infant botulism happens when spores germinate in the infant's intestines and produce toxin there. Constipation is the earliest sign, then poor feeding, weak cry, floppy baby syndrome. Some studies suggest infant botulism may be responsible for up to 5% of sudden infant death syndrome cases. In November 2025, the FDA expanded a recall of ByHeart Whole Nutrition infant formula after infant botulism cases were linked to it. The case fatality rate for foodborne botulism remains at 5 to 10% even with treatment. Respiratory failure, multiple organ dysfunction, sepsis from secondary infections. No true antidote exists. The antitoxin stops circulating toxin from binding to more neurons. But it cannot reverse damage already done. Shigella causes bacillary dysentery, shigellosis. Only 10 to 100 bacterial cells are needed to cause infection. Compare that to other bacteria that require 1,000 to 100,000 cells. Shigella spreads via the fecal-oral route. Contaminated food, water, hands, direct person-to-person contact, sexual contact, changing diapers. The bacteria survive stomach acid. They reach the large intestine. They invade epithelial cells on the basolateral side. They reorganize the cytoskeleton, induce actin polymerization, and get endocytosed into the host cell. Once inside, they multiply. They spread cell to cell without ever leaving the protected intracellular environment. The inflammatory response damages the epithelium, degrades the epithelial barrier. Bloody diarrhea ensues. The World Health Organization estimated in 2005 that shigellosis occurs in at least 80 million people globally and results in about 700,000 deaths per year. Most cases occur in developing countries. Young children are most commonly affected. In the United States, about 450,000 Shigella infections occur each year. Antimicrobial resistant infections result in an estimated $93 million in direct medical costs. 4 species exist. Shigella dysenteria, Shigella flexneri, Shigella boidae, Shigella sone. In the United States, Shigella sone is most common. Shigella dysenteriae and boidae are rare but continue to be important causes of disease in areas with fewer resources. Shigella dysenteria type 1 can be deadly. It produces shigatoxin, the same toxin that E. coli O157H7 produces. The toxin enters host cells by endocytosis and terminates protein synthesis. The cells die. Hemolytic uremic syndrome can develop. Among shigellosis cases worldwide, approximately 20 to 119 million illnesses and 6,900 to 30,000 deaths are attributed to foodborne transmission. Foodborne transmission has been reported among travelers in multiple outbreaks. Tourists became ill after eating contaminated foods in hotels, on airplanes, on cruise ships. Symptoms usually start 1 to 2 days after exposure. Diarrhea, often bloody, fever, abdominal pain, tenesmus, the feeling you need to pass stool even when your bowels are empty. Symptoms last 5 to 7 days, but it can take several months before bowel habits return to normal. Bloodstream infections occur in 0.4 to 7.3% of shigellosis patients, most common among those with weakened immune systems. HIV, diabetes, cancer, severe malnutrition, children. Bloodstream infections are most commonly seen with Shigella flexneri and Shigella dysenteria. Patients with bloodstream infections are at higher risk of death compared to those without. Generalized seizures have been reported among young children, usually with high fever, low blood sugar, or abnormal blood electrolytes. Reactive arthritis develops in about 3% of cases. About 0.4% to 7.3% develop hemolytic uremic syndrome when the infection involves shigatoxin-producing strains. Use occurs when antimicrobial resistant drugs are used to treat the infection or when treatment starts after 4 days of symptoms. It is the leading cause of death in Shigella dysentery outbreaks. The bacteria spread easily in daycare settings, poor hand hygiene, inadequate sanitation. Gay, bisexual, and other men who have sex with men are at increased risk due to sexual activity involving stool exposure. People experiencing homelessness face high risk when shigellosis spreads in the community. About 25% of sporadic cases in the United States are travel associated. International travelers to Africa, Central America, South America, and Asia face the greatest risk. Resistance to former first-line antibiotics is widespread. Ampicillin, cotrimoxazole, nalidixic acid. Resistance to quinolones has been increasing. International travel is associated with a 6-fold higher odds of infection with quinolone-resistant Shigella. Reduced susceptibility to azithromycin and ciprofloxacin has been documented among men who have sex with men in several countries, including the United States. Now, Vibrio vulnificus, this one lives in warm seawater, brackish water, estuaries, coastal areas. It thrives when water temperatures exceed 18 degrees Celsius, when salt concentration ranges from 2 to 25 parts per thousand. Hurricane season creates perfect conditions. Heavy rains, flooding, warmer waters, the bacteria multiply. 2 ways to get infected. Eat contaminated raw or undercooked seafood, especially oysters, or expose an open wound to contaminated water. Vibrio vulnificus is the leading cause of death related to seafood consumption in the United States. It causes 2 distinct syndromes: primary septicemia from eating contaminated seafood, or necrotizing wound infection from water exposure. The case fatality rate for primary septicemia exceeds 50%. For wound infections, about 15% overall to mild, but rises to 54% in patients with underlying liver disease. After a 75-year-old patient suffered boat-related hand trauma after Hurricane Ian in Florida, he developed hypotension, altered mental status, pain, swelling, and erythema of the left hand. Necrotizing fasciitis. Intravenous antibiotics and fluids started immediately. The infection progressed rapidly. Emergent surgery required. Vibrio vulnificus confirmed. The patient survived but required extensive debridement. In coastal hospitals, they know to expect these cases during hurricane season. Wounds acquired in marine environments, lacerations, punctures, abrasions. The bacteria colonize quickly. Intense skin reaction, cellulitis with blistering bullae, untreated. It proceeds rapidly from skin lesion to necrotizing fasciitis with myonecrosis C, and then overwhelming sepsis. Primary wound presentation is seen in about 25% of all Vibrio vulnificus cases. Overall wound infection mortality approximates 25%. For primary septicemia, you eat the contaminated oyster. The bacteria enter through the small intestine. They reach the bloodstream. Sepsis develops within hours. Fever, chills, hypotension, skin lesions appear, hemorrhagic bullae, large fluid-filled blisters, ecchymoses. The skin turns purple, necrotic patches, multiple organ dysfunction syndrome sets in early. Without aggressive treatment, death follows quickly. Liver disease is the biggest risk factor. Cirrhosis, hepatitis, hemochromatosis, elevated iron levels, diabetes, immunosuppression, chronic kidney disease. These conditions increase susceptibility and severity. In Taiwan, the aquatic environment and endemic hepatitis contribute to high susceptibility. A multidisciplinary treatment protocol uses ceftriaxone or ceftazidime combined with doxycycline or minocycline, followed by emergency fasciotomy and intensive care unit admission. Between January 1996 and December 2011, 100 cases were reviewed. 18 patients died, 18% mortality. Unknown injury events, presence of multiple skin lesions. Leukocytes, less than 10,000 cells per cubic millimeter. Platelets, less than 100,000 per cubic millimeter. Serum creatinine, 1.3 milligrams per deciliter or higher. Serum albumin, less than 2.5 milligrams per deciliter. And delayed treatment beyond 3 days post-injury were associated with significantly higher mortality. Treatment delayed beyond 3 days is an independent factor, indicating poor prognosis with an odds ratio of 10.75. In a 3-year prospective study at a tertiary coastal hospital, 184 consecutive necrotizing fasciitis patients were evaluated. 20 died. Mortality rate of 10.9%. Vibrio species caused 40 cases. Staphylococcus aureus caused 31 cases. The death group had significantly higher incidence of shock at emergency room and bacteremia. Necrotizing fasciitis caused by Vibrio vulnificus is more rapidly progressive and fulminant than MRSA necrotizing fasciitis. Surgery must happen fast, within 12 hours of admission. Studies show that delaying surgery beyond 12 hours significantly worsens outcomes. Fasciotomy, debridement, necrosectomy, in severe cases, amputation. The bacteria spread through fascial planes at astonishing speed. You can see the infection advancing hour by hour, red streaks, swelling, blisters forming, skin blackening. The tissue dies. Mechanical ventilation may be required for weeks. Climate change is expanding Vibrio vulnificus range, warming ocean temperatures, rising sea levels. The bacteria now appear in waters farther north than before. Cases have been reported in cold climates. In Binzhou, China, a coastal city, a 61-year-old male with chronic liver disease and diabetes presented with chief complaint of pain in both lower legs. No other symptoms initially. Within 12 hours of arrival, typical skin lesions appeared. He developed primary sepsis. Despite prompt appropriate antibiotic and surgical treatment, he died 16 days after admission. The infection occurred in winter. Vibrio vulnificus does not just strike in summer anymore. Multiple antibiotic resistance has been found in up to 50% of Vibrio infections. Third-generation cephalosporins, tetracyclines, carbapenems, fluoroquinolones work in vitro, but resistance is spreading. The CDC recommends combination therapy. Intravenous ceftazidime with either ciprofloxacin or doxycycline. There is a soil sample from a scaffold site in Dublin dated 12/21 that contains DNA matching known plague strains. Wait, different bacteria, but same principle. The bacteria sit there, dormant, viable, waiting for conditions. Vibrio vulnificus sits in warm, brackish water during hurricane season. Salmonella sits on raw chicken in every grocery store. Listeria hides in biofilms on processing equipment. Campylobacter waits in that drop of chicken juice on your cutting board. E. coli O157H7 lives in cattle intestines and transfers to ground beef. Staphylococcus aureus lives on the hands of food workers. Shigella passes through populations with 10 bacterial cells. Clostridium botulinum spores survive boiling and wait in improperly canned jars. In 2024, the FDA investigated 26 outbreaks. The USDA investigated 7. The CDC coordinated investigations of 13 multi-state outbreaks caused by E. coli, Listeria, and Salmonella, plus 17 to 36 investigations of foodborne illnesses each week. Most outbreaks do not get identified. Most cases do not get reported. People get sick. They recover. They never know what hit them. About 95% of foodborne illnesses occur sporadically in individuals rather than in identified outbreaks. The CDC relies on voluntary reporting from public health authorities. A person gets sick. Maybe they seek medical help. Maybe the doctor orders a stool test. Maybe the lab cultures the sample. Maybe they identify the bacteria. Maybe they send the isolate to the state lab for typing. Maybe the state reports it to the CDC. Miss any step and the case vanishes from official counts. The 48 million illnesses per year estimate is probably conservative. The real number could be much higher, because for every reported case, there are 20 unreported cases. For every hospitalization counted, there are dozens of people sick at home. For every death attributed to foodborne bacteria, there are deaths where the cause was never identified. Someone elderly with multiple health conditions dies. Sepsis listed on the death certificate. No one cultures the blood. No one traces back to the potato salad at the family gathering 3 days before. The bacteria get a free pass. In late 2024, Castile, ground beef traced to multiple states was pulled from shelves when routine testing picked up E. coli O157H7 contamination. No illnesses reported. The recall happened before people got sick. That is how the system should work. But it relies on testing. It relies on monitoring. It relies on enforcement. In March 2025, the National Advisory Committee on Microbiological Criteria for Foods was eliminated. The National Advisory Committee on Meat and Poultry Inspection was eliminated. Surveillance systems like FoodNet remain as of April 20th, 2025. But expect more foodborne outbreaks going forward as safety systems get reduced or dismantled. The bacteria do not need help. They already have advantages. Antibiotic resistance is spreading. Campylobacter resistance to quinolones, Shigella resistance to azithromycin and ciprofloxacin, Vibrio vulnificus resistance to multiple agents, some Salmonella strains resistant to ceftriaxone. The treatments that use to work are failing. Bacteria acquire resistance through plasmids, through horizontal gene transfer, through selective pressure from antibiotic use in agriculture and medicine. The resistant strains spread. A person infected with resistant Campylobacter travels internationally, sheds bacteria in stool, contaminates food or water in another country. Someone else gets infected. They travel. The resistant strain circles the globe. 25% of sporadic Shigella cases in the United States are travel-associated, 60% of Shigella dysentery infections, 44% of Shigella boydii infections. Travelers pick up resistant strains abroad and bring them home. Biofilms protect bacteria in processing plants. Listeria forms biofilms on stainless steel. Those biofilms can persist for years if not properly addressed. Standard sanitizers do not always penetrate. The bacteria sit there, protected in drains, on equipment, releasing cells into the food stream. A single contaminated slicer can inoculate thousands of pounds of deli meat. Cross-contamination in commercial kitchens happens constantly. Raw meat on one cutting board. Ready to eat salad on the same board 10 minutes later after a quick rinse, or a knife used to cut raw chicken, then rinsed and used to slice bread. The bacteria transfer, temperature abuse during transport, a refrigerated truck breaks down. The food sits at 50 degrees Fahrenheit for 6 hours. Bacteria multiply. The truck gets repaired, the food gets delivered. Temperatures are back to safe levels when it arrives. No one knows. The bacteria are already there in sufficient numbers. Home kitchens are not better. Most people do not know safe internal temperatures. They judge doneness by color or texture. Chicken that looks done might still have Campylobacter inside. Ground beef that looks brown might still have E. coli in the center. Pork chops that seem cooked through might not have reached 145 Fahrenheit. Thermometers are not used. Cross-contamination happens constantly. Raw meat juice on hands. Touch the faucet. Touch the refrigerator handle. Touch the salt shaker. The bacteria spread. Sponges and dish towels harbor bacteria. A damp sponge at room temperature is a perfect incubator. Wipe down the counter after handling raw chicken. The bacteria are now on the counter, in the sponge. Use the sponge on dishes. The bacteria transfer. Leave food out at room temperature. The 2-hour rule, 4-hour cumulative rule. No one tracks. Leftovers sit out after dinner while people socialize. 3 hours later, they go in the refrigerator. Bacteria have multiplied. Immunocompromised populations are growing. HIV, cancer chemotherapy. Organ transplant recipients on immunosuppressants. Inflammatory bowel disease on biologics. Elderly populations increasing. Very young children, pregnant women. These groups face higher risk and worse outcomes from the same bacteria. Listeria targets them specifically. Vibrio vulnificus thrives when liver disease or diabetes is present. Salmonella bacteremia happens more often in immunocompromised patients. Campylobacter can cause relapsing infections in those with antibody deficiencies. Shigella bacteremia occurs more commonly with HIV, diabetes, cancer. The case fatality rates in these populations are much higher than in healthy adults. Food recalls happen after people are already sick. The Boar's Head Listeria outbreak. First case in late May. Recall in late July. People were eating contaminated deli meat for 2 months. 10 people died. The Yu Shang food outbreak. Products distributed before the contamination was identified. 3 babies died. The McDonald's E. coli outbreak. Onions served from mid-September. Recall on October 22nd. 1 person dead. 4 with hemolytic uremic syndrome. Dozens hospitalized. By the time the source is identified, the damage is done. The lag time between initial contamination, first illnesses, outbreak detection, source identification, and recall means people keep eating contaminated food even as investigators work. Many illnesses occur weeks or months after recalls are announced because consumers do not know about the danger. The alert systems are inadequate. No single method reaches everyone who purchased a product. There is no vaccine for any of these bacteria. Vaccine candidates exist for Shigella in various stages of development. Live attenuated, conjugate, ribosomal, proteasome vaccines. None licensed yet. The World Health Organization has made Shigella a target for vaccine development. Natural immunity does develop after exposure. But vaccines take years to develop, test, approve. Manufacturing capacity needs to be built, distribution systems set up. In the meantime, the bacteria spread. No vaccine for Salmonella for human use. No vaccine for Campylobacter. No vaccine for E. coli 157H7. No vaccine for Listeria. No vaccine for Vibrio vulnificus. Prevention relies on food safety practices, proper cooking, avoiding cross-contamination, hand washing, temperature control, safe water sources. Those practices stop bacteria at the individual level. But system-level prevention requires surveillance, enforcement, inspection, testing, recalls, all of which depend on resources and political will. In a 2024 study of American military personnel deployed in Thailand, over half of those with diarrhea were infected with Campylobacter species. Travelers get hit hard. Shigella, Campylobacter, Salmonella, contaminated water, street food, inadequate sanitation. Travelers' diarrhea is often bacterial. Most people take antimicrobials prophylactically or start them at first symptom. That drives resistance. A study in Australia and Canada found that 40 to 50% of locally diagnosed shigellosis cases were travel associated. People travel, get infected, return home, spread resistant strains. The bacteria flow across borders constantly. Climate change affects bacteria ranges. Vibrio species expand north as waters warm. Longer warm seasons mean longer windows for contamination. Hurricane intensity increases. More storm surge, more flooding. More brackish water intrusion into areas that were freshwater. More Vibrio exposure. Sea level rise affects oyster beds. Oysters filter huge volumes of water. They concentrate bacteria. One contaminated oyster can contain enough Vibrio vulnificus to cause lethal sepsis. Extreme heat events affect food storage. Power outages. Refrigeration fails. Food sits at unsafe temperatures. Bacteria multiply. Then power comes back. Food goes back in refrigerator. Looks fine. Bacteria already grew. Drought affects irrigation water quality. Less water available. More concentrated with pathogens. Produce washed in that water gets contaminated. Heavy rainfall events cause agricultural runoff into water sources. Animal waste. Fertilizers. Bacteria. That water irrigates fields. The bacteria get on produce. Surveillance matters. FoodNet tracks 8 pathogens in 10 states. About 16% of the UIS population. Campylobacter, E. coli, O157, Listeria, Salmonella, Shigella, Vibrio, Yersinia, Cyclospora. Active surveillance means laboratory-based case finding. When a clinical lab identifies one of those pathogens, FoodNet collects detailed information. Patient interviews, food histories, clinical outcomes, strain typing. The data feeds into outbreak detection. If multiple cases with the same strain appear across different states, an outbreak investigation starts. PulseNet compares DNA fingerprints. When patient samples match food, samples match, the source is identified. Recalls happen. That system depends on labs doing cultures, reporting results, saving isolates, conducting typing, entering data. It depends on epidemiologists interviewing patients, tracing food sources, coordinating across jurisdictions. It depends on FDA and USDA inspectors sampling products, tracing distribution chains, inspecting facilities. It depends on CDC coordinating and analyzing data. Break any link and the system fails. Reduce lab capacity, cut epidemiology staff, eliminate advisory committees, reduce inspection frequency, stop monitoring. The bacteria do not stop. They keep doing what bacteria do: growing, producing toxins, contaminating food, infecting people. The question is whether humans detect and respond. In the past, cholera killed millions until John Snow traced it to the Broad Street pump. Typhoid fever spread until Typhoid Mary was identified and isolated. Polio paralyzed children until vaccination programs started. Those diseases required surveillance systems, public health infrastructure, scientific investigation, and intervention. Bacterial food poisoning is no different. The bacteria are there. They're not going anywhere. Human systems either control them or they do not. You opened this script thinking food poisoning meant a bad night. Now you know. Paralysis from botulinum toxin. Kidney failure from Shiga toxin. Necrotizing fasciitis from Vibrio vulnificus eating your flesh. Meningitis from Listeria. Guillain-Barré syndrome from Campylobacter. Hemolytic uremic syndrome from E. coli. These are not theoretical risks. These are confirmed cases with dates, locations, patient counts, death counts, documented in public health records from 2024 and 2025. 1,392 people sickened in 2024 from contaminated food. 487 hospitalized, 19 dead. Those numbers doubled from 2023, and those are only the reported cases. The ones where someone got sick enough to see a doctor, got tested, had the pathogen identified, got reported to health authorities, the real number is 20 times higher, maybe more. In a Florida hospital after Hurricane Ian, a patient arrived with necrotizing fasciitis. Vibrio vulnificus, surgeons debrided necrotic tissue. The infection had spread through fascial planes. Hours mattered. Delay meant death. In Fresno County, California, 8 people at family gatherings ate home canned cactus. Botulism, intensive care, mechanical ventilation, weeks in the hospital. All survived because they got antitoxin fast. In 19 states, people ate boar's head deli meat. 10 died from listeria. In 14 states, people ate McDonald's. Quarter pounders, 1 died from E. coli, O157H7. 4 developed hemolytic uremic syndrome. In 34 states, people ate cucumbers. 551 got salmonella, 155 hospitalized. The bacteria do not care about brand names or quality ratings or health scores. They care about temperature, moisture, pH, oxygen, nutrients, time. Give them conditions and they grow. The bacteria win unless you understand them. Cook ground beef to 165, whole cuts to 145 with 3-minute rest. Poultry to 165. Use a thermometer. Wash hands with soap and hot water for 20 seconds before and after handling raw meat. Use separate cutting boards for raw meat and ready-to-eat foods. Clean and sanitize surfaces between tasks. Refrigerate perishable foods within 2 hours. 1 hour if temperature is 90 or above. Keep refrigerator at 40 degrees or below. Freezer at 0 or below. Thaw food in refrigerator, not on counter. Do not eat raw or undercooked eggs. No raw milk. No raw oysters if you have liver disease or compromised immunity. Avoid cross-contamination. Do not use the same utensils or plates for raw and cooked foods. Wash produce thoroughly. Cook home canned foods by boiling 10 minutes before eating. Pressure can low acid foods. Follow tested recipes. Do not taste food to check if it is spoiled. If in doubt, throw it out. That protects you individually. System-level protection requires functioning surveillance, inspection, testing, enforcement. It requires researchers studying virulence factors and resistance mechanisms. It requires public health infrastructure. It requires laboratories that culture samples, identify pathogens, conduct typing, share data. It requires epidemiologists who investigate outbreaks, trace sources, coordinate responses. It requires regulatory agencies that inspect facilities, mandate recalls, enforce standards. It requires a political system that funds those activities and does not dismantle them. When those systems function, outbreak curves peak and decline. Source identified. Product recalled. Cases stop. When those systems break down, outbreaks spread unchecked. More people get sick. More people die. The bacteria keep circulating. This script covered 8 bacterial pathogens: Salmonella, Listeria, Campylobacter, Escherichia coli, Clostridium botulinum, Staphylococcus aureus, Shigella, Vibrio vulnificus. Each one has specific mechanisms. Each one requires specific conditions. Each one causes specific disease syndromes, but they share common threads: low infectious doses for some, ability to survive outside hosts, resistance to environmental stresses, ability to produce toxins, capacity for horizontal gene transfer and antibiotic resistance acquisition, targeting of vulnerable populations, and reliance on human behaviors for transmission, contaminated food, unsafe water, poor hygiene, temperature abuse, cross-contamination. These bacteria are endemic, meaning they are always present in animal populations, environmental reservoirs, human carriers. Elimination is not realistic. Control depends on breaking transmission chains at multiple points. A USDA report from January 2025 found persistent violations at multiple meat processing plants, sanitation failures, temperature control failures, inadequate record keeping. Those violations create conditions for bacterial contamination. A Food for Thought report from February 2025 documented that Listeria recalls rose from 47 in 2022 to 65 in 2024. Salmonella recalls rose from 21 in 2019 to 41 in 2024. The trend is wrong direction. More contamination, more recalls, more illnesses, more hospitalizations, more deaths. Part of that reflects better detection. Genomic sequencing allows outbreaks to be identified that previously would have been missed, but part reflects actual increases in contamination events. Processing plants with inadequate controls, distribution chains with temperature failures, products reaching consumers while contaminated. The bacteria are not going to stop. They are successful organisms that have been around for millions of years and will be here after humans are gone. They occupy ecological niches. They have evolved mechanisms for survival, transmission, and pathogenesis. They acquire resistance to antibiotics. They adapt to new environments. They hitchhike on global trade and travel. They benefit from climate change. They benefit from intensive animal agriculture with crowding and antibiotic overuse. They benefit from inadequate sanitation infrastructure in developing countries. They benefit from lapses in food safety in developed countries. They benefit every time humans fail to follow basic hygiene and temperature control. The evolutionary pressure is on the bacteria's side, but humans have advantages too. Scientific understanding of pathogenesis mechanisms, molecular typing to track strains, surveillance systems to detect outbreaks early, communication networks to coordinate responses, regulatory authority to mandate recalls and shut down contaminated facilities, treatment modalities like antitoxins, antibiotics when they still work, supportive care like mechanical ventilation and dialysis, vaccines in development, food safety guidelines based on decades of research, the capacity to educate populations on safe practices. Those advantages only work if they are maintained and used. Surveillance requires funding. Labs require trained staff and equipment. Investigations require time and expertise. Enforcement requires authority and will. Treatment requires healthcare infrastructure and access. Education requires accurate information and effective communication. In 2024, the recorded increases in foodborne illness severity were concentrated in 13 outbreaks, 12 caused by Listeria, Salmonella, or E. coli. Those 13 outbreaks drove hospitalizations from 230 to 487, drove deaths from 8 to 19, 13 outbreaks. But those 13 outbreaks affected over 1,000 people. Boar's Head alone hit 61 patients and killed 10. The system failed at the processing plant level. Violations documented for 2 years. Production continued. Contaminated product distributed. People died. The USDA launched an internal investigation into its inspection and reporting processes. That investigation should find systemic problems, because the outcome indicates systemic problems. A processing plant with documented violations for 2 years should have been shut down. It was not. Product kept moving. The bacteria kept infecting. Final data point. In Europe, 36 countries reported foodborne and waterborne outbreaks in 2018. Staphylococcus was among the top 10 pathogens for hospitalizations. In China, Staphylococcus ranks third for food poisoning incidents. Campylobacter causes 9 million cases per year in the European Union. The problem is global. The bacteria are global. The solutions require global coordination, surveillance, reporting, information sharing, rapid response. That happens through WHO, through bilateral agreements, through scientific networks. It happens when countries prioritize public health over short-term economic considerations. It happens when the political will exists to maintain the infrastructure. When it does not, people die from eating contaminated food.